Hello and welcome back to day 2 of Advanced Therapies Connect. I hope you all had a good rest after what was most definitely a very busy day yesterday. If you missed any of yesterday's sessions, don't worry, they will be available to watch on demand for another 30 days right here on this platform. And similarly, today's sessions will also be available to watch for 30 days starting from tomorrow. Now, before we jump into our first session, we have a quick poll for you, which should be appearing on your screen around about now. The team would be very grateful if you could respond with whether you would consider traveling to Miami for the conference live in June. Okay, our first session today is a panel discussion hosted by Ology Bioservices, which will focus on the practical decisions in process development. We have experts from AVROBIO, Notch Therapeutics, Amendo and Dark Horse Consultancy who'll be joining Ology Bioservices to dig in to some of those challenges. So let's head over to our panel. And first, our moderator, Jared.
Ology Bioservices is a US-based biopharmaceutical contract development and manufacturing organization providing cGMP and non-GMP high-quality services to our clients for the development and production of cell and gene therapies, viral vectors, oncolytic viruses, and vaccines against infectious diseases and cancer. Learn more at www.ologybio.com.
Thank you, David. Today's panel discussion is on practical decisions in process development. Basically, what can you do today that will make your process more robust? My name is Jared Denham. I am here representing Ology Bioservices, where I am the VP of Manufacturing and the site lead in our new California site. For this panel discussion, I have 4 of my friends in the industry to discuss about process development, considerations for process development, as well as case examples from therapeutic product developers. Before I introduce the panel here, I'd like to talk about a little bit about what Ology Bioservices is on the next slide. And I would like to introduce Ology Bioservices to the Facilitate community. Ology Bioservices is a CDMO that is headquartered in Alachua, Florida. In that facility in Alachua, Florida, it's a very, very large facility, 183,000 square feet, where they are currently producing vaccines and traditional biologics for government contracts as well as commercial operations. Ology is currently expanding into the cell and gene therapy space. Because we make live viral vaccines in Florida, we are able to easily manufacture in vivo gene therapy products, and we are currently producing some viral vectors in Florida. In California, where I work, we are building a cell and ex vivo gene therapy capability in Alameda, California. Prior to Ology Bioservices, my background is in process development, manufacturing, quality, regulatory for the cell and gene therapy space. I have over 20 years of experience working initially for 3 different product developers, making cell and gene therapy products. I worked in 3 biotech companies in the Bay Area and San Diego, California, basically working in increasing roles of responsibility in those companies. And then for the last 5.5 years, prior to starting in Ology, I worked for a consulting firm that specializes in cell and gene therapy. That is Dark Horse Consulting, where I was a principal and worked on probably over 60 or so different clients in the cell and gene therapy space. I was asked to do this panel discussion and was able to bring on some of what I thought were groups that had a tremendous amount of knowledge with regards to the cell and gene therapy space, primarily in CMC and process development and analytics, especially. So with that, on the next slide, I'd like to talk about a little bit about what today's session is going to be. So again, today's session that we're here today is really about practical and applied process development strategies for the cell and gene therapy products, with examples from industry. Initially the panel discussion will talk about some considerations with regards to process development of cell and gene therapy products. And then we'll move into 3 case examples where we will have industry representatives present a little bit about the challenges and in their technology with respect to both process development and analytical development. So the format for today's session is, we're going to have short presentations from each of 4 panelists. Each presentation is pretty short, so it's about 5 minutes, so total of 20 minutes, and then followed by that, after everyone has presented their information, then we will have some questions. Those questions can come from you, the audience, as well as other, the panelists on the session. And then, of course, I probably have some questions as well. And so we're going to use those questions to have a really great discussion on process development for the cell and gene therapy industry. So before we introduce the all of the panel, I think that this is a good opportunity to take a snapshot in time with respect to process development analytics. I think that, this industry is moving very, very quickly at the speed of light. There's a tremendous amount of technology tools that are available out there. So, these are just examples, but, hopefully you, the audience, can get some information to build your process to be more robust. So on the next slide, I'd like to use this slide to introduce today's panelists. So this is the order that these people will be presenting. First off, we will have Sarah Mills, Senior Consultant from Dark Horse Consulting, give, basically high-level survey of the landscape with respect to what Dark Horse sees with respect to process and analytical development, what to do, what not to do, some key considerations, to build your program. Next up, we'll have Azadeh Ghalipur, VP of Manufacturing Operations at AVROBIO, to present her team's ex vivo autologous lentiviral vector gene therapy for lysosomal diseases such as Fabry, Gaucher, and other rare diseases. And then next we'll have Hadass Shoham Nissan, Program Manager for Amendo Biotherapeutics, headquartered in Israel. And speaking from Israel, again, another autologous ex vivo, this time gene-edited cells for severe congenital neutropenia. And then last but not least, Emily Titus, VP of Process Sciences from Notch Therapeutics, where they are developing iPSC-derived T cells for a variety of diseases. So with that, I would like to hand it over to Sarah Mills from Dark Horse Consulting, where she's going to present, a high-level landscape with respect to process and analytical development.
So it's nice to be with everybody today. I would like to start by just saying thank you, Jared, for the opportunity to be here. And I am quite humbled to be on the panel with the other individuals that have that you're going to be hearing from. So today, I want to talk about maintaining forward momentum and courage in the process development space for cell and gene therapy. My background is in cell therapy, so much of what I discuss will come with some color from cell and gene modified cell therapy. So if you advance to the next slide, where we're going to start our conversation today is actually not going to be in process development, but in product development. Before we talk about process development, we have to align on product development. And there are two kind of high-level categories that I want to discuss. Number one is upfront, what is incredibly critical for your ability to do process development in a successful manner is defining what your product is. The tool that we consistently use and guides our clients in many different ways is QTPP, so a Quality Target Product Profile. This document can be revised over time and can grow with your product as you develop through your preclinical and into your clinical manufacturing. And this document outlines items such as process description, product components, dose strength, route of administration, container closure choices, and of course, critically quality attributes and your drug product specifications. And the other element I'd really like to, invite this group to think about is embracing the concept of what we call a minimum viable product. Okay, so the figure on the right, what you see is a semi-pithy Venn diagram, which has two circles, one that says minimum, one that says viable. And while minimum may be inferior products that nobody wants to end up using, and viable may end up having all the bells and whistles, where you want to be, especially as an early startup company, is in the middle. Okay, and that is minimum and viable. These products have features that are required for you to go out and test a market hypothesis. However, it allows you to build small first, and then on top of that, start to build and build and build. And the overall saying that we use a lot internally, and I like to use with my clients, is don't let the perfect be the enemy of the good. Okay, next slide. So advanced therapies process development really starts when we talk about this saying that you'll hear a lot, and this may be semi-controversial, but the process is your product. That adage is really not helpful in many ways, and it may actually be harmful from the perspective of taking hold within a development program in a given company. And what it's done is it's really led to the avoidance of changing process throughout a development cycle, and those cumulative challenges that you end up facing, which are inevitable to occur as you go through a development cycle, end up being either avoided or everyone's scared to address them. And then it can really end up causing challenges as you get into your later phase clinical trials and you're faced with the absolute necessity to perform things like comparability studies. Okay. The other thing that's important to note is that your process will change, and your non-clinical and pivotal material that you manufacture, while it doesn't need to be the process that you go into the clinic with, it does need to be representative of that process. Okay, so you have to be very thoughtful about the changes in which you plan on performing. The most important consideration, or at least one of the most important considerations, in my opinion, is understanding your cost of goods early. Okay, so your COGS will help identify where you should put investment in your process development. A perfect example of that would be potentially of what not to do, would be transduction. While transduction is a critical unit operation for many gene-modified cell therapies, it's not necessarily a labor-intensive day. And while it may require a significant amount of vector, spending time optimizing a little bit here, a little bit there on a unit operation, which ends up not having a lot of overhead on top of the actual vector, may end up not turning around into high cost benefits, especially when you start to consider the development cost and the comparability study that you may have to incur if you're already within your clinical manufacturing cycle. Next slide. So to support these changes, which will inevitably occur during your process development cycle, it's really important to have a robust routine characterization and release testing panel. There are there's this idea that over time, through a drug product development, that you should basically be paring back this panel. Okay, so early in development, maybe even preclinical, you should have many, many analytical tests that you're using to characterize your process in many different ways, individual in-process testing, and a large panel with your final drug product to continue to characterize your material as you develop. And once you get into the clinic, consider what is absolutely critical to have release criteria on and set minimal release criteria. Then throughout the bulk of your product development and process development activities, start to remove some of those characterization tests that were either found to not be informative or incredibly difficult to manage for one reason or another, and move other tests that were maybe deemed as just characterization tests that have ended up being incredibly value added, move those to be part of your release criteria. And then obviously, as we get towards the end and we look at commercial development, a fully developed specification for your drug product would be where you want to end up leading to, that has this very robust history of data behind it. The other thing I want to encourage this group to really consider, and I may be a broken record at this point, is that do not delay your focus on potency, okay? Assess multiple potency test methods and think early about what your potential mechanism of action is. This is incredibly critical. And the industry has provided us with multiple cautionary tales from 2020 of what happens when potency in MOA is not fully developed or considered early, okay? Along with, thinking about this multi-layered characterization moving into release testing, the other critical element that you want to consider is the orthogonal or assay matrix approach to these tests, especially potency. And to support all of this data that you'll be collecting throughout your process development cycle is building in a robust tracking and trending program alongside. And that will allow you to understand your process and then turn the knobs once you have a better understanding of what your process is telling you and what changes cause what effects. Lastly, I've alluded to in the last few slides, but I'd like to talk about on the next slide is comparability. So let's start planning for comparability and stop trying to avoid it, because it's inevitable and it's something all or most drug product development programs are going to have to go through. Okay, it is not, as we said towards the beginning of this talk, it is not necessary to develop a commercial process for your early clinical trials, but you should always be thinking about what you want that process to look like from early on. And to do that, you're going to start planning when those transitions in your process should occur. And this figure that you see on the screen basically is just a very crude and rudimentary demonstration of how as you move from research all the way through clinical into your post-approval activities, the burden of comparability increases. So being thoughtful and about when you perform comparability and what elements you want to put into that comparability study will really benefit your program overall. There are common comparability failure modes that I'd like us to discuss just really quickly, which include the lack of upfront communication with your regulatory authority. Go get buy-in before you execute on a study when you can. Over-promising. Be thoughtful about what you say you're going to do, and don't overpromise and promise the world, because they will say, great, let's see it. And other items such as inadequate analytical test methods. So this goes to the maturity of your test methods and when you're using them and how you demonstrate, if it's site-to-site comparability, how the test methods are performing the same. And the most important thing, and I cannot stress this enough, is having sufficient retains. A retain program that follows you throughout your development life cycle is one of the most critical and valuable resources you can build alongside your process. And one last thing to consider before I hand it off to my colleagues is depending on your product type, you may want to consider a comparability program. So if you have multiple donors, if you're an allogeneic cell therapy where you're going to be going back to the well of different donors often, or you're you're a stem cell based therapy and you're going to have multiple WCBs, then what you could consider is proposing that comparability program as part of your regulatory filings instead of having to constantly have a formal interaction with the regulatory authorities as a way to get buy-in prior to performing those. And lastly, just as a reminder to this group is what, some very common misconceptions about comparability, which includes comparable is not identical, okay? And that's what I want to leave you with. Comparable is not identical. They are two separate things. And thank you for your time. I look forward to the questions, Jared.
Thank you, Sarah. That was a great, thoughtful, and important presentation. Sarah and I actually go way back. We've worked with each other at two different companies, most recently at Dark Horse, and those were very important words. Thank you. Next up, we have Azadeh Ghalipur from AVROBIO. Again, another friend of mine that I've known for quite some time in the industry, and she's going to be presenting practical decisions in manufacturing and talking a little bit about AVROBIO's Plato platform. Azadeh?
Hello, and thanks for having me today, Jared. I'm Azadeh Ghalipur, Vice President of Manufacturing Operations at AVROBIO. AVROBIO is a clinical stage company striving to halt or reverse disease with one single dose of ex vivo gene therapy. Next, please. As a publicly traded company, I want to note this presentation may contain forward-looking statements. We refer you to our most recent SEC filing for more information. Next, please. Here is our ex vivo lentiviral gene therapy approach, which likely looks familiar to many of you in the field of cell and gene therapy. Right where our company started, our team recognized that in order for our product to reach patients globally, we need to automate this process. I'm going to walk you through the practical decisions we made early on in order to establish a robust manufacturing platform, which we call Plato. Next, please. Plato is unique for many reasons. It's our commercial-ready, end-to-end production platform featuring our novel plasmid system, optimized vector production, and closed and automated drug product manufacturing, and more. Plato is currently in clinic for two of our lead indications, Fabry and Gaucher disease, and cleared by multiple regulatory agencies for use in clinic. Next, please. Plato has been a major breakthrough. The fully closed system greatly reduces risk of drug product contamination, meaning we can operate in lower class clean rooms. Automation reduces variability, which allows us to manufacture across the globe without losing consistency. Cell selection, transduction, harvesting, and cryopreservation are all optimized and automated, and we have delivered industry first in this area. The Plato platform is designed so that when we move from one indication to another, we only need to swap the vector. Everything else remains the same. We believe that using a single platform will reduce regulatory burden for each new program, save cost, time, and reduce complexity as we grow our pipeline. Next, please. Plato is built to solve the key manufacturing challenges. I'm going to talk through two of these today. Next, please. 5 years ago, when we started AVROBIO, most lentiviral vectors were manufactured in adherent culture. The challenge for us was that using the adherent platform would not allow us to serve a global patient population. To address this challenge, we decided at the onset to develop a scalable suspension platform. We are currently best in class for industry lentiviral vector production at 200-liter serum-free suspension culture. So let's talk about our suspension platform and see how it's doing. Next, please. Here are the vector titer results from multiple batches across 2 different AVROBIO investigational programs, Fabry and Gaucher. The titers from both production processes are well above the industry standard of 10 to the 8th. A high titer means higher yield of vector and subsequently fewer batches required to fulfill demand. That then translates into fewer rounds of production, less time, and lower cost. Now, let's look at the scalability. Next, please. As we think about a growing pipeline, the scaling capacity is critical. This starts with our proprietary optimized plasmid. Each of our product candidates utilizes the same 4 plasmid system, where 3 out of the 4 plasmids used in every vector are the same. This provides economies of scale for plasmid manufacturing and also reduces complexity in terms of characterization. We believe this will be helpful in achieving regulatory comfort with the vector design. The vector itself can be mass-produced well in advance and then put into long-term storage for later use. To scale the drug product, we add manufacturing suites and automation units based on demand. Next, please. Everything I told you today about, it's doable. Sure, it will be a lot of work. In some cases, you may need to tweak or optimize existing technologies or develop new ones. But overcoming these manufacturing challenges, including reaching a global population, are possible to achieve, and similar solutions can be applied across many settings. As you develop production platforms for new therapies, always have the end in mind. Start early, and as Sarah just told us, invest in analytics. Thank you.
Awesome, Azadeh. Thank you so much for presenting on AVROBIO's platform. I'm getting a reminder from outer space right now. Reminder for the audience to ask questions to the panel in the box next to the session, and we will answer those as best as we can. We have 2 more presentations, and then we'll get into the questions and discussion. So I'd like to transition to Hadass Shoham Nisan, who is another friend of mine working at Emendo Bio, and introducing Emendo Bio to probably the larger cell and gene therapy audience. So with that, Hadass will be speaking on their ex vivo autologous gene-edited cell therapy for severe congenital neutropenia and other diseases. So thank you, Hadass, for presenting today.
Thank you, Gerald. A quick overview about Amendo. Amendo was founded in January 2016 and recently acquired by Anges, which is a Japanese cell and gene therapy company. We're R&D focused with 70 employees, which most of them are located in Israel. We're in our preclinical development stage. We have multiple programs for ex vivo and in vivo, and an extensive IP portfolio. Amendo has 2 technology platforms. One is the discovery of novel CRISPR-associated nuclease, and the second is the optimization of these CRISPR nucleases. Next slide, please. So how can we advance CRISPR from a revolutionary research tool to a level of precision and efficiency that will enable us to target any genetic disease? So Amendo has its Omni technology platform. We have more than a dozen novel CRISPR nucleases that were discovered in-house, and we have proprietary advanced protein engineering to make these nucleases to be able to target novel gene editing strategy like allele-specific editing. Next slide, please. So most of the genetic diseases are caused by a dominant condition. such as compound heterozygous, gain of toxic function, and haploinsufficiency. In order to treat these diseases, one allele needed to be added in, while the other one need to be intact. Such specific editing requires meeting the highest bar of specificity, and the discrimination between the allele needs to be on a single base mismatch. Currently, for most genomic targets, there are approaches have not demonstrated the specificity, discrimination, and the activity needed to achieve this specific editing, which leaves us with a huge unmet need. The novel omninucleases wishes to take these nucleases and for challenging diseases that are not addressable with the current generation of the CRISPR technologies. Next slide, please. So the first indication we are going for is the allele-dependent severe congenital neutropenia. This is a dominant mutation caused by protein misfolding and maturation arrest. To date, there are more than 200 unique allele mutations that has been identified, and this is an ultra-rare designation with approximately 60,000 patients worldwide. The unmet need of these patients is high. They're using daily injections of GCSF, and which has severe side effects and increasing the risk of AML and MDS. The current treatment is the allogeneic hematopoietic stem cell transplant. This is a curative treatment, but the success rates are very low. So in Amendo, we wish to take the autologous, the patient autologous hematopoietic stem cells and to collect them and isolate the cells for CD34 plus cells, and then using ex vivo editing with our own specific Cas nucleases and 2 single guide RNA to edit these cells based on our SNP-based editing strategy that enable us to address more than 80% of the patient. We expect that a low level of correction will be sufficient, and with regard to the clinical protocol, we have clear and short-term clinical endpoints. Next slide, please. So how do you accelerate the development but keeping a robust process? So before starting the development program at Amendo, we took few decisions. The first one was that the development process will be done in-house. This way, the know-how and the process control will be kept at Amendo. Also, with regard to critical raw materials, which I already mentioned that they are proprietary of Amendo, they are developed in-house. The omninucleases it has higher activity and the higher specificity with regard to the current nucleases outside. And the guide RNA are unique for each SNP strategy, and we have succeeded to develop a shorter guide that will enable us easier and robust guide RNA production. We're going to use closed and automatic system, which give us lower chances for contamination, and it will be easier from the technology transfer to a CMO and even for the employee trainee. The product will be cryopreserved for easier logistic and longer stability. And this will enable us to use a central CMO versus a local or a site-specific CMO. During the process development, we are also performing in vivo experiments in critical steps of the process, and in order to verify that the in vitro results are similar to what we see in vivo. And with regard to the drug product manufacturing for the CMC and the toxin biodistribution, we have decided that we will also perform these studies in-house, and therefore we started to apply GLP standards in Amendo, and this will also provide us a shorter timeline to clinical manufacturing. The only open question at the moment is whether we want to go for a CMO, qualify a CMO, or whether the Amendo team will perform GMP clinical manufacturing for clinical products. Next slide, please. And this is the Amendo platform. We have several programs for ophthalmology, skin and CNS, and also partner programs. And thank you, Jared, for having me today.
Thank you, Hadass. I actually know of a CDMO. It's in California. I work there. No.
Let's talk.
Yeah, definitely. Yeah, thank you for that information. Really excited for Amendo.
Thank you.
Yeah. So our last presentation, we're switching gears a little bit. This is allogeneic iPSC-derived T cells from Emily Titus, Notch Therapeutics. Hi, Emily. Thank you for joining us today.
Hi, Jared. Thank you. I'd like to thank you for inviting me to speak on this panel with a very impressive panelist today. I think I was chosen because Notch is an earlier stage allogeneic iPSC company. My name is Emily. I'm Vice President Process Sciences at Notch Therapeutics. Next slide. So as Jared mentioned, Notch Therapeutics is developing T-cell products from stem cells, and in particular, we're focused on development from induced pluripotent stem cells. We're a new startup founded at the very end of 2018, based on two technologies from the Sunnybrook Research Institute and University of Toronto. Our scientific founders were Juan Carlos Zuniga-Fluker and Peter Zanstra, who are world leaders in their respective areas of developmental immunology and bioengineering. And our platforms are really based on their deep understanding in stem cell and developmental biology, bioengineering, and advanced cell manufacturing that the founding team was brought together around. So we've grown quickly. We're currently 35 people with 3 sites in Toronto, Vancouver, and Seattle, and we are continuing to build our capabilities and our team over the coming years. Next slide. So Notch's capabilities in T-cell differentiation are enabled by our core platform technology, which we call the engineered thymic niche. This is the system that allows us to differentiate stem cells to T cells in a fully defined and feeder-free system. So the engineered thymic niche is essentially the presentation of DL4 and VCAM on a surface, and the presentation of those proteins to stem cells causes Notch signaling to be upregulated and the cells to differentiate to the T lineage. This has previously only been accomplished by using feeder cells, such as the OP9 system, expressing DL1 or DL4 on the surface of the cells. So this is the first example that we know of a fully defined system. And the technology can be used in either an adherent system, which we call generation 1, or as coded on a microbead, which we call generation 2. And we're really focused on the development of the second generation of the ETN, because this will allow us to culture the cells in suspension and achieve a scalable system for future cell production needs. What we've demonstrated is that the addition of VCAM synergizes with the DL4 and accelerates the kinetics of the differentiation of the T cells. And the other advantages of this microbead format is that we can control the timing of the addition of the bead-bound ligands and the removal to really fine-tune the Notch signaling throughout the differentiation process and achieve different T cell lineage subtypes out of our process. We're currently focused on the development of CAR-T and TCR products, mature effector T cells, but our platform has the future potential to be modulated to produce other T lineages, such as regulatory T cells. Next slide. So I wanted to give you a snapshot of how the differentiation process progresses from our starting material, which is a gene-edited iPS cell, through to a CD34 expressing cell, at which point we add the engineered thymic niche to the system to cause the differentiation to the progenitor T cell lineage marked by CD7 and CD5 positive cells, before which the cells are in specialized expansion and maturation media to result in a mature CD8 single positive T cell subtype. And I've tried to outline the tremendous expansion that we see throughout this process, which allows us to envision a initial scale of production that would allow us to create about 100 doses from a 1 liter bioreactor of iPS cells. So what I think is important to note in this cartoon at the bottom is that we can imagine producing a single batch of a large number of doses of cells. And we have already demonstrated that we can do the initial stages of this in a stir tank bioreactor, and that we can achieve the co-culture of the engineered thymic niche and the differentiating cells in a number of different types of vessels. And our approach here will be to use a system that can be scaled up and down, so that even our early research material will be produced using a system that we believe will be representative of our future scaled process. Next slide. And finally, we have built a systems biology and computational modeling team to help support our process development efforts. So even in the early days, as we begin to learn more about our culture system and the marker expression of the differentiating cells along the way, we want to collect all of that information so that we can learn from it. And the modeling team has produced a few very important models for us. One is a model of T cell differentiation, and another is a model of notch signaling, so that we can understand how the engineered thymic niche controls the cells during differentiation, and so that we can, again, learn from the data that we collect in understanding our process better. Next slide. So thank you, Jared, for allowing me to show an insight into the work that is coming for Notch over the next few years, and just a note that we are hiring in the next year and growing. Thanks, Jared.
Thank you, Emily. That was a great presentation of Notch Therapeutics and your technology. So it looks like we have about 15 minutes, a little shorter, for the questions and discussion. Again, to the audience that's out there, feel free to ask questions in the panel, and I'll take them up. They're right here on my. They're going to come up on my cell phone, so I'll look at those. But in the meantime, maybe I can start off with some questions, and we can get a discussion around. This is, I might ask a specific question of someone on the panel, but if you are another panelist that have additional information or thoughts, please feel free to chime in. So, one of the things that I, have learned in my years of experience working on a variety of different products is, as you're developing a process, you really need to think about risk, okay, and what you're going to do, and I know that we didn't really. This is not a topic that came up in any of these presentations, but I do think that as you're developing a process, depending on what phase of development you're in, you have to do a series of risk assessments. And I do know somebody who knows a lot about risk assessments, and that's my friend and colleague Sarah Mills from Dark Horse. I know that they work with a variety of clients, and also are always assessing risk, based on their development. So I'd like you to kind of open up the panel and the discussion, Sarah, with your thoughts and kind of what Dark Horse sees with risk assessment, when and how, and where does a company start, that's just building a technology from the ground up? When do they start assessing risk?
Yeah, that's a great question. And I am the self-deemed master of disaster, which is my favorite self-made nickname that I gave myself at Dark Horse. So there's a lot of different methodologies that can be used in risk assessment. There's a lot of different tools in our tool belt, and they are very specific but can also be very flexible. Okay, so early in development, a lot of times you'll use more simple, maybe like a 2-factor risk assessment, which will look at it'll make you a little matrix that will then allow you to assess risk from maybe a process development point of view, like I spoke about during my slides, or just an overall product development view from 2 different factors, just like it says in the name. So you could look at urgency. Okay, how fast do we need to address this specific need or this specific gap? And then severity. So if we don't address this, what is the worst case scenario of what could happen? So always kind of assessing things from a worst case scenario and then determining what the mitigation strategy is. My other favorite tools are RAM. So RAM matrix is very common and is used throughout, and especially in later development. That's something you will see quite a bit. And then my other favorite, which takes some time, I think, to really fully understand and implement in a meaningful way, but failure modes and effects analysis, FMEAs, whether it be for a process from an end user point of view, or also as a tool in one of my favorite kind of subcategories, which is raw material qualification activities. So using an FMEA to ensure that you have put together the right hazard control strategy to use some of these unique materials that maybe aren't made at a GMP, at a GMP level, and how you can take those and still translate them into material which is appropriate for use in the clinic.
Great, thank you. Anyone else on the panel have any thoughts on risk assessment or planning, up front, to evaluate risk, or thoughts, related to mapping out changes and what risks you're willing to take? Does anyone else have any comments or thoughts? Okay. So, looks like I'm going to ask some questions. Feel free, guys, to add in and ask your own questions as well. But I'll go ahead and move to maybe Emily, which I thought was a really great presentation. Kind of outlined, what you guys are planning. I know that you're early stage, you're growing. Just if you could provide a little bit more detail. Maybe a little bit more details on what you're doing with regards to, your iPSC derivations. I know that there's probably a whole bunch of work with regards to cell banking. What are you doing to ensure that these cell lines and banks are really fit for purpose for essentially over the life cycle of any product line that you develop, hopefully into the commercial space. I'd love to hear your thoughts, kind of a little bit more details, planning and designing with, ultimately the end in mind.
Yeah, that's a great question, Jared. And it was certainly one of the first decisions that we had to make about where would we source our iPS cell line from. When you think about that iPS cell line, that becomes the basis of your product going into the clinic, and if everything goes well, forward into your commercial product. So that was a very important decision for us. We felt that it was an important investment to create our own iPS cell line. Historic lines have been made using research grade materials or otherwise have improper or incomplete documentation. And we thought that it was worth the trouble to us to invest in the creation of our own line, where we would have an understanding of the donor selection process, the donor consent and testing documentation would be, available to us to understand before we started the process. And we could understand as well from a business perspective, the technology that was being used, the licensing requirements of that technology, and other aspects of how the documentation would be prepared during the manufacturing of that iPS cell line. So we felt strongly that we wanted to put that in motion for ourselves. And it took, a good CMO partner, as well as, a better part of a year to get ready for that, that process. But as you mentioned, this cell line, if successful, will be the basis of our product. And to further elaborate on that, it's not just the initial iPS cell line, but every time we put an edit into that cell, it increases the value of that cell to us, and the cost involved in going back to the beginning increases. So just the more that we can do to understand the quality of this iPS cell line, the less chance we have to repeat work later on in our development. Does that help, Jared?
Yeah, absolutely. Great. I mean, you and the team at Notch are really, I believe that you're really setting yourself up for success by investing early, into these initial cell lines and banks, and I think it's a great strategy. It's great that you're early, that you can do that, and I'm glad to see it. It's something that I've actually been wanting to see for the last 20 years, is somebody's to start investing early into their initial starting materials. So thank you.
Jared.
Yeah.
Can I ask Emily a follow-up question?
Sure.
So Emily, that was great. I'm wondering whether you think the field will move to having commercial ready cell banks, iPS cell banks. So to your point, not for every earlier stage company to spend a year time and resources to develop that initial starting material. Do you think there is a movement in the field? Curious to hear your thoughts on that.
Yeah, I'm not sure. I think Sarah could have some input into this question as well, from more of a regulatory perspective of whether that is doable or not. I haven't seen kind of that done successfully. I think every product is very unique, and we wanted to make very specific decisions for the purpose of our product. And every donor, the donor itself was something that we wanted to consider the traits and features of that for the eventual product that we created. Mm-hmm. So we didn't spend a lot of time considering whether we could find something off the shelf for some of those reasons. But perhaps Sarah, you have other insights into if that could be possible.
Yeah, I think, Azadeh, that it is probably the most common trend to do what Notch is doing, which is go make your own, just because then you have control, and that control over not only the information you have up front, and how thoughtful you can be about the manufacture of that critical starting material, but then also the initial time investment, I understand, is kind of what ends up being this, we have clients that are looking for something off the shelf because they don't want to take the time. Yeah. But by the time they perform proper due diligence on an off the shelf line, you're almost to the time in which you would be to have your own line completed, right? So I think that from my perspective, I think that Notch is kind of going in the right direction as far as making their own, and then, putting all of that thought and then having control. But there are some groups that are working to have these off the shelf options. Mm-hmm. And it'll be interesting to see how those play out as far as where the fight is to, do you get some kind of limited use in X or Y, Z direction, and what differentiation you're going to perform, et cetera, et cetera. So it's a very thoughtful question.
Thank you for that good discussion. We have probably enough time to discuss what I think is another important factor with regards to process development, something that a couple of groups touched on, a couple of people touched on in their presentation, and that's with respect to analytical test methods. I personally believe that you can't do good process development without having established good analytical test methods. So I'd like Azadeh and Hadass to speak a little bit. Azadeh, and then just shortly thereafter, Azadeh, keep it short. Hadass, I'd like to just be interested in, you guys are thinking, to accelerate the development of your robust process. I'd like to know your thoughts on how you're going to incorporate analytical methods into your plans. So Azadeh, go ahead and answer with respect to AVROBIO's analytics.
Yeah, absolutely. And I try to be brief here. So absolutely, Jared, I think it's extremely important to invest in analytics and invest in analytics early. It's important, I think, for everyone who is listening to really think through whether or not the analytic method that you're using is going to be qualifiable and validatable at the end. I give you one example. Percent transduction. All of us are using it. This single cell colony culture for many days, trying to pick, I don't know, hundreds of colonies, and then trying to assess percent transduction that then really becomes a very important attribute, sometimes a critical quality attribute for your drug product. So think about platforms that are out there. Sequencing has moved really since years ago, and there are platforms out there that utilize sequencing in order to identify rare cells. Those same technologies can be used in the field of cell and gene therapy to measure percent transduction. So I'll leave it at that, pass it to Hadass.
Thank you. Hadass, so can you tell us a little bit about more of Amendo's analytics?
Yeah, I first of all, I totally agree with Azadeh about the analytical methods. But in Amendo, we are trying to keep to go for the acceptable analytical methods with what we can. We don't want to invent the wheel.
Yeah.
So we are trying to keep. We do have a unique raw materials for each of our indications. So these complex things a bit, but we are trying to keep it simple with regard to analytics. But if we're talking about for a second about the raw materials which are unique. We do, except for the analytical, need to do a biological activity test before we are releasing such a unique raw material. So this is also an important issue with regard to raw materials.
I can see that. Thank you. So it looks like we're getting close to the end of our session. I wanted to really thank all of the panelists for joining me very early in the morning on the West Coast, but really excited for these 4 companies and their technology and their development. And hopefully we were able to give you kind of a snapshot of what different companies are doing out there. So again, thank you to everyone for joining us. With respect to me and my new venture, these are the types of companies that Ology Bioservices would love to serve in Alameda. So this is my shameless plug, but feel free to reach out to any of the members of the panelists with additional questions. I'm sure that they will take the time and be thoughtful in their response. And feel free to contact me as you need further development and contract manufacturing in the cell therapy and ex vivo gene therapy space. So with that, I'd like to turn it back over to David. And thanks a lot for your time.
Thank you.
Thank you very much.
Bye-bye.
Oh, bye-bye, everyone.
Bye-bye.
Thank you very much, Jared. Thank you very much indeed to the entire panel there. Up next here on the network track, join the coffee hangout discussion asking what is the biggest bottleneck on the road to digital transformation. These will be informal discussions like you would have over a coffee at a live event. After the coffee hangout, we have 2 Meet the Expert sessions alongside a roundtable discussion on using automation as a solution to the workforce development challenge. So lots to look forward to. Hope to see you back here soon.